Balaji to coordinate the technical session. As we proceed further, I request everyone to once again keep their phones on silent or vibration mode. Our first speaker is Nick and Daphne Maulis from Bahamas, and next is Dr. Jaysri Das Sarma. Dr. Balaji, please take over the mic. Thank you.
Good afternoon, everyone. Good afternoon. I would request everyone to kindly take their seats. Okay. Thank you, everyone. After a very wonderful inaugural session, now we are going to the technical session. The technical session 1. This has invited lectures. There are 2 speakers in the technical session 1. The first lecture will be by Nicholas and Daphne Mollis, and second will be by Dr. Jaysri Das Sharma. And to conduct the session, I request Dr. Venkatesh Devanu to be the chair for the session and Dr. Pushpavalli to be the co-chair for the session. The first lecture will be presented here and second one will be online. Thank you, Dr. Venkatesh Devanu. Thank you, Dr. Pushpavalli.
Good afternoon. Welcome back after nice snacks and coffee and chai. We have now technical sessions going on. I'm happy here Dr. Balaji has introduced session. My co-chair is Dr. Pushpavalli. My name is Venkatesh. I invite now Nick from Abaco NEEM from Bahamas. We have a golden couple from Bahamas. I call them golden NEEM couple. They're producing NEEM premium chemical-free NEEM products for health, skin care, garden care. They're located in Abaco. They offer tools and ship worldwide by DHL. They feature product like neem oil, pest repellents, neem soaps, and various kind of products. Thank you for coming all the way from Bahamas to Pune, and I request you to take over the next presentation, please. Thank you.
I just want to have, the first word. My husband, of course, will have the last word. And together, we will do our best. Let me see. I think I have to stand over here because I'm so short, nobody can see me.
And please, 25 minutes you can take perfectly, no problem.
Yes, thank you. So we have a slideshow that is going to show you images while we're speaking, because they may be more interested than looking at us, of our home, of our farm, where we live, so you can have an appreciation for our environment, because as you know, we are all products of our environment. So I'm going to turn over now to Nick.
All right, thank you for this opportunity. Our journey started over 32 years ago, planting our first seeds in the ground in October 1994. And today, we are standing before you making this presentation. NEEM brought us together and has afforded us an incredible life experience. It all started at a botanical show in Gainesville, Florida, when I came across 2 gentlemen handing out NEEM flyers about a neem tree from India that offered multiple health benefits. Through my affiliation with the University of Florida, I used their library to do further research, and after learning that NEEM was a natural preventive that can potentially help to prevent and manage many health issues, in particular diabetes and hypertension, both of which were prevalent in our country. My gut feeling told me that this would be a great investment and can potentially benefit the healthcare in our country. With the help of Mr. John Conrick, the gentleman who introduced me to NEEM, we imported over 8,000 NEEM seeds from India. And once the seeds germinated successfully, we applied to the government for agricultural land and took out a 21-year lease. Today, 32 years later, we have 130 acres cultivated with over 7,000 neem trees. Also, 1,200 variety of coconut trees and over 107 variety of fruit and flowering trees. Within 3 to 3.5 years after planting the first neem seedlings in the ground, we commenced the production of neem oil, neem seed cake, neem leaf capsules, neem extract, salve, and an insect repellent. Later, we developed a variety of soaps, lotions, cream, lip balm, and pet products. We encouraged organic farming practices and used the neem oil cake as a soil enhancer, which we all know now is a natural fertilizer. We distribute to local vendors throughout the Bahama Islands and ship internationally on our online website sales.
Now, after 30 years, we've created a fully integrated tree farm that operates completely off the grid. We are powered solely by the sun and for over 12 years and utilize fresh groundwater. Our water is second to none. We sit atop a huge natural reservoir. In fact, we do no irrigation on our farm. The trees are feeding right out of the high water table. We have also right now we're evolving into a destination that appeals to bird watchers, natural lovers, nature lovers, both domestic and international visitors, and are now developing housing on the farm for stayover visitors. We host school tours where we hope to inspire potential future farmers to an industry that presently depends primarily on seniors. Like ourselves, it's imperative that we encourage young people to get involved in this industry. The Bahamas' main industries are tourism and finance, and we partner with our Ministry of Tourism, offering group tours and events, and our ambassadors in a program called People to People in our country. In 2025, we were awarded Best Commercial Farm of the Year by our Ministry of Agriculture. Finally, they're recognizing people in agriculture. NEEM has not only provided a valuable source of income, but has also allowed us a great lifestyle that promotes our personal health and that feeds our soul. I can honestly say that it's thanks to being NEEMatized that we have reached our 70s in excellent health and totally non-medicated. Our real joy is that of spreading the knowledge of NEEM and all of its amazing health benefits with others. This is why when we were approached by Dr. Martin to participate in growing a neem plantation on his ranch in Paraguay, we embraced the opportunity. By providing over 140,000 neem seeds from our certified organic farm and participating as consultants with our knowledge and experience for over 6 years, we were able to facilitate the cultivation of the second certified organic neem farm. We encourage collaboration between businesses across the globe to ensure that continued development and promotion of NEEM for the betterment of mankind at large. We need to hold hands. We need to partner with each other. We need to share information.
Health is wealth, and the better each one of us does, the better we all do. The good life is about learning to live in nature without destroying it and feeding off of what the land provides for us. Simple living, surrounded by nature, breathing fresh air that the neem tree helps provide to filter, and sharing with others has brought a sense of fulfillment and enrichment in our lives. We are truly grateful for the opportunity to help others improve their lives through the miraculous benefits of the neem tree.
We want to especially thank the NEEM, World NEEM Organization for pulling all of us together, those who are here and those who are watching from abroad. Thank you for the honor of receiving the Eminent International Persona Award. As the first organized neem plantation in the world, we might not be the largest, but we are certainly helping to maintain the standard and quality of neem oil and its byproducts. We know that in our country, we have contributed positively to the health care of our little nation, and that was the main original purpose of starting the Abaco Neem Farm in the Bahamas. We welcome all of you to visit, maybe not all at the same time, and see and experience our farm, and to visit our beautiful country, the Bahamas. And now my husband will have the final word.
Thank you. With this lifestyle, we have evolved into natural healers. We don't have the doctor titles, but we believe in integrated medicine. Several doctors in our country are now starting to catch on. As we all know, nothing good happens overnight, but we see a bright future for NEEM, especially for preventable diseases such as diabetes and hypertension. We would like to emphasize how very important it is for keeping proper records and sharing information. The WNO is so important, providing us an opportunity to hold hands and to share information. We all need to support it because holding hands and collaborating provides accessibility for others and spreading the word of NEEM. Healthy competition provides quality growth. This should not be all about financial gain. This should be about the improvement of mankind's health. Take care of nature, and nature takes care of us.
And of course, as a woman, I have the prerogative of changing my mind. And I just want to say a very special thank you to Dr. Kurtari for being an excellent host, and to all of your team for being so gracious and for this opportunity. It's truly our honor. If anyone has any questions, we'd be happy to answer them for you.
Any questions?
They come all the way. I think there's something, definitely we have something to in question and answers. Yes, please, Dubey. Introduce yourself.
Go ahead.
How much seed or the fruit per kg you get from per tree you get from the tree?
It all depends on the size of the tree, as you know. So we live in a hurricane zone. As we all know, the neem tree can grow over 60 feet. Some species grow 90 feet. All right? So, but we have to prune our trees year round. The tree is family to the mahogany tree. It's not going to sway in high winds. So if we harvest couple hundred pounds from each tree, that's our capabilities, all right? And we also harvest by hand. We do not harvest off the floor, all right? We try to produce a product that's taken orally and to keep as close to a pharmaceutical grade as possible. All right? So we do use our byproduct of the neem oil cake. We feed our sheep. We put that in their food. As we know, it's a natural dewormer for parasites, etc. It's also an enhancer for them. All right? So, but to answer your question, on our size, we originally had 10,000 neem trees. But back in 2019, September 1st, 2019, we had a terrible Category 5 cyclone hurricane. We lost over 3,000. So since that time, we have incorporated more mango trees, more avocado trees, citrus trees, fruit trees, flowering trees. Our farm is evolving into almost like a botanical garden. We have created a habitat for animal life and a place for people like yourselves to come and visit and disconnect from this fast life. All right? But the bottom line is, to answer your question, we can comfortably produce about 10 tons. We only harvest what we need, all right? But if we had the opportunity, if somebody wanted, we can supply. But there are many big companies here, present here, that I think are doing a wonderful job. Okay.
Yes, introduce yourself, please.
Yeah, I'll just, am I audible?
Yeah.
Yeah. It was a wonderful presentation and a talk by you. I am Dr. Tarun Kant. I am from Arid Forest Research Institute, Jodhpur. Been working on NEEM myself for a long time now. My question is, looking at your journey of how you have introduced NEEM and the way it has thrived in your land. Just wanted to know, when you started putting in the NEEM seedlings, what was your seed source? Was it a known seed source that you sourced your seedlings or maybe your saplings from? Was it from your local stuff, or you got it from around the world? Number one. And number two is, the NEEM has a very good regeneration potential. So when new NEEM seeds are falling down, they are naturally must be regenerating. What do you do with the natural regeneration that is taking place in your developed forest of NEEM? Thank you.
To begin with, we got our original seeds from India. Right? And as you know, they have a survival rate of maybe 2 weeks. Out of that original 8,000 plus seeds, we had a survival rate of 10%. But after 3 years, we had our own NEEM seeds, and we slowly expanded. Nothing good happens overnight. And with the excess seeds that we do not collect, they enhance our soil. We have our soil, we're lucky to have 3 to 4 inches of topsoil. We have a lot of limestone. But I can tell you, the large trees, around the large trees, the soil is beautiful. You know, some parts of the world, they call NEEM the fertilizer tree. It definitely deserves that name. Everything around the NEEM trees grows in harmony. Our soil, after 32 years, has been enhanced because of the NEEM trees. All right? Go ahead.
I just want to explain the reason why we only had 10% germination was because the seeds got held up in U.S. Customs, and so we lost time. And going, you know, the Bahamas, they didn't know, America didn't know what a NEEM was, didn't know what a NEEM seed was. So it took some convincing, and thank goodness Nick had a book about NEEM with him. So he had to actually convince the customs agents that this was a legitimate seed coming from India. So that's one of the reasons. The other thing also is, because we mentioned about hurricanes, when the trees go down, we are able to examine the soil around those roots, and that's how we know how that soil has been enhanced by those trees. Otherwise, we are digging in rock. And we have to build our own soil. So we mulch, we compost, we are constantly building soil to grow and plant trees. We are not looking to do huge harvest or mass production. We are looking really for a quality of product and for a lifestyle that supports us without too much stress. We're not looking to be the biggest. We're creating a botanical a health retreat where we invite you to come and bathe through the neem forest, connect in nature, disconnect from the gadgets, and improve your health.
And what is interesting, our dream is, as we talk to you, we're building accommodations so researchers and scientists like yourselves could come and visit the farm. And what a wonderful place to study and to do research on the Abaco Neem farm. We encourage that. We welcome.
Actually, two of our members have been to the farm. Susie from the UK and Klaus Furlow, I think you would know about him from Canada. They have both come and visited with us on the farm.
Any other questions? Did we answer your question? Thank you. Any
On the
other
back,
questions?
please. Come to mic, please.
First of all, very good afternoon to everyone. So my question is like, how much of...
Introduce yourself, please.
Yeah, my name is Bala from Neemco Traders and Neem Life Global from Namakkal district, Tamil Nadu. So like my question is about like, how much amount of neem cake can be feed to the animals or else for the cattle?
Okay, for example, with chickens, we start out with a very small amount, all right? Okay. We use the neem oil cake, very little, but we use it consistently twice a week in their water. And we can tell by the energy of the chickens, their egg production, we can see by their mobility. I believe in visual inspection. A lot of people tell me, why are you always mowing in the field and working in the field? Because I believe in visual inspection. I believe in keeping records. Every night, Daphne can tell you, I sit in my rocking chair and I write down what I learned that day. Every day we step foot on the farm, you learn something. Every day. All right, so we feed it to our sheep. We also have sheep. All right, and so we feed small amounts to our sheep. But in the pastures, in the paddocks where the sheep feed, there are neem trees. And the animals know when they're not feeling well what grasses to eat. And so we see them eating on the lower leaves of the branches. And our sheep are very, very healthy. They gain weight. We have no issues with their eyes, no intestinal issues. And we are very satisfied with the potential for neem with animal husbandry. Very satisfied.
Fine. Another thing, so is there any sweetening agents is to be added in the neem cake to feed the sheep or else for the cattle?
Yes, but you start off with small amounts.
No, no.
Any sweetening agents? No, not really, no.
No.
None at all.
We don't need to have sweet. Even our children shouldn't be getting sweet to be encouraged to eat.
But
Sugar is not good for us.
But here I have tried for feed to the cows. But while I'm trying to feed the cow using the neem cake, the cow is not supporting to eat. So that's what I need to.
You might be adding too much at one time.
All
So
right.
you introduce slowly.
Okay.
Okay, introduce very slowly. Start with small amounts, and then visual inspection, record keeping, and as your herd increases, you might want to increase.
Okay. Okay. Another one thing, it's about like neem bark. How we can use the neem bark in these like what kind of products we can use? We can make it and all.
Okay, so all good health starts with dental hygiene. So we incorporate the neem bark along with baking soda, only those two ingredients, and for dental hygiene, we get great results. We've worked with some dentists for surgery using, the neem oil and the neem leaf extract, but we certainly believe in the neem bark. We also use it in our tincture because, as you know, neem has a very bitter taste. So we add some of the roots and the neem bark to that tincture.
Okay,
All right? Start always with small amounts and then you graduate.
fine. Thank you.
Anybody else there with questions, please? Introduce yourself, please. Wait a minute. No, they have. You have question. Sorry.
Sir, I want.
One more. Introduce yourself, please.
Actually, I want to know.
Introduce yourself, please.
Thank you, sir. How?
Introduce yourself.
Sir, I'm Thanga Kumar. We are using NEEM in the textile industry for making antimicrobial textiles. So my question is that after your 30 years, now in your country, how many people is doing this same? I want to know. I'm very curious in knowing about your efforts. Yeah, after 30 years, you are promoted in your country. Many people are doing this. These NEEM-based products.
As you know, most people are slow to change. And in our country, we are very much influenced by North America that uses a lot of synthetic fertilizers. So in our country, we have a very, very shallow water table, very close to the surface. We have some of the best drinking water in the world. And water is becoming a precious commodity. So we are teaching the farmers. We go to farms. We also encourage farmers to come to our farm, all right? And they can see the results. You know, there's a saying, don't tell me, show me. So we like to display and encourage. Farmers are catching on. But to be quite truthful with you, as we travel around the world, we see agriculture on a decline. And where you see agriculture on a decline, you see those people's culture on a decline. So we are trying to encourage our government. I think they now understand, because of food security globally, that agriculture is a very important economic base. And how to farm, incorporating the neem cake, is very, very important. And we see a great future for those of you that are creating the neem cake. The market is unlimited. We encourage you.
Thank you. Thank you very much for taking the seed from India and promoting it. Thank you very much. Thank you.
Yes.
Thank you.
Hello. Thank you. Thank you, Mr. Nicholas and Daphne. So we congratulate for your effort for taking NEEM from India to establish in your place. And you also explore a lot of the opportunities for NEEM, especially in veterinary area.
And I have to thank India for bringing NEEM to us.
Okay. Yeah. Thank you.
Thank you very much. Now we have the next session is from Dr. Jayashree Das Sharma. It's an online event. She is a professor in the Department of Biological Sciences, Indian Institute of Scientific Education and Research, IISER, Kolkata. The online presentation. Are we ready with that? Yes, over to you, our team, IT team. Thank you.
Shall I start? Shall I start, sir?
Yes, start please, yes. Go ahead.
Can you hear me?
Very well, yes.
Okay, thank you. Thank you for your kind introduction, and good afternoon, everyone. And my sincere thanks goes to the organizers for giving me an opportunity to present our work, what we are doing for last 6 to 7 years at our Institute IISER Kolkata. So I'm going to talk about a broad spectrum therapeutic perspective of neem bark extracts and its bioactive compounds from viral defense to oncology. So first of all, I would like to say that the slides are not moving. The slides are not moving. Hello? The slides are not moving. Yes, thank you. So we all know that modern medicine has its own complications, like, several complications. I'm not going to talk about it much due to paucity of time, but the side effects of the modern medicine. And we need of nowadays a holistic therapeutic approach. We can think about it from the ethnomedicinal plant, and especially which provides the multitary benefits. One of such, there are quite a few plants which are used in India, especially for its ethnomedicinal values like aloe vera, tinospora, ashwagandha, and our research is mainly focused on neem as a reductive indica, because neem has many pharmacological effects, and it has antimicrobial, anti-inflammatory, antioxidative, wound healing, insecticide and neurohepatic properties, as well as anti-diabetic properties and anti-cancer properties. But not much is known about its antiviral properties, as well as the mechanism how neem bark or its extract or its bioactive compound can work is not very much known. Though there are a large number of compounds are available, like, azadirachtin, nimbin, nimbolite, these are mainly available commercially, but it is very difficult to procure because it's expensive and we don't know how to apply it to a clinical aspect. So we need to have the multidimensional benefits from these products, bioactive compounds, as well as whole neem bark extracts with negligible side effects, non-toxic for humans and safe for environment. So first of all, I would like to convey my gratitude to my grandfather, Sri Vedanto Bachishpati Sri Narayan Krishna Sengupta, who actually inspired me when I started working on the cancer and virology field. He told that neem bark extract is not much explored and it may have some ethnomedicinal properties, so you should look about it. So my sincere gratitude goes today. So today, whatever I'm going to show it to you, the inspiration came from him. And this work actually culminated when SARS-CoV-2 came, because 2017, this work started to take shape when Dr. Lakshmi Sarkar, who was then PhD student, she had a botany background. She wanted to work on neurovirology. So we thought it will be a good opportunity for her to explore the role of neem bark extract and its anti-neuroviral properties of neem bark and its compounds. So that's what we started the work in 2014-15. But when it is culminated for during her research tenure, that time SARS-CoV-2 came, and IUSSTF told us that you should do a collaborative work between India and US, as I have a long-term collaboration with US, and I'm also adjunct faculty at University of Pennsylvania. So large group of people from India under the leadership of myself and Professor Debnath Pal at ISC Bangalore and Karolinska Institute, Dr. Utpal Niyogi, Professor Arup Banerjee from RCB and Dr. Parna Gupta from ISR Kolkata, we made a team and we joined hands with several faculties like from Emory University, University of Pennsylvania, Miami University, and especially the University of Colorado. And we made this work possible. And now I'm going to share some of the clips of that work. So our work is mainly in the mutant beta coronavirus, like which belongs to the beta coronavirus family, like SARS-CoV and SARS-CoV-2. And SARS-CoV-2 is a BSL-2 pathogen now, but when it came due to its zoonotic potential, it was considered as a BSL-3 pathogen, and biosafety level 3 is not possible to do it in the laboratory. So most of the work is dependent on the idea or the literature we generated through a prototype beta coronavirus, which is the virus I'm working throughout my postdoctoral tenure, and also when I got the independent faculty position at Thomas Jefferson University and in India, I'm using this as a model system. And by using this model system, understanding the mechanism of how the virus causes the disease has led to a roadmap for designing the therapeutic strategies and the aspect we study neuroimmune modulation, cell-cell communication, genomic control of pathogenic properties, structure function analysis of the murine beta coronavirus spike protein and host cell response. And now it gives me a understanding of how we can design the antiviral therapeutics. So when SARS-CoV-2 came, due to our extensive experience with the murine coronavirus, we were requested to write several reviews to shed some light in understanding the neuropathogenesis of the CNS pathobiology of the SARS-CoV-2, where we the help from the SARS-CoV-2 spike protein and compared it with murine beta coronavirus spike protein, and we observed that it has a major determinant of neuropathogenic properties. And also, we took it like a pleiotropic anti-cancer, antiviral, and anti-immunomodulated role of methanolic neem bark extract. So by combining these, this is a model I should say we are working on these, like we have a cause-effect relationship because we give the virus to the brain and we get acute stage encephalitis, meningitis, perivascular coughing, microglial nodule, and during chronic stage it causes demyelination and axonal loss. Long time we know this virus-induced neuroinflammation is a very good model to understand the very common neurological disease of the young adults in US, UK, and New Zealand, Netherlands, Australia, Northern Canada, but it is not that much prevalent disease in India, or maybe it is not diagnosed. And one of the property of that is like, it causes demyelination, and pathological hallmark is demyelination and axonal loss. That's what is a very good model system to understand that. So we targeted one of the property is the viral fusogenicity, as it is very much important for viral spread. So we wanted to see whether we can use the neem bark extract and we can see whether it can reduce the viral titer, like, it cannot infect cell to cell. So we did fusogenicity study, as well as when we treated with neem bark extract, we observed there is a significant low viral titer, and by EGFT tag viral, we showed that the virus is not spreading from cell to cell. But in that, we took it further to understand how actually that can cause. So we showed the in vitro assay by luciferase, and we showed that there is a significant less viral fusogenicity. So MNB restricts spike-mediated entry and replication in neuro-2 cell, which generally get infected with the virus. And we took it, we did a lots of studies, which due to paucity of time, I'm not showing, but you can see in our paper. And then we took it for in vivo studies, and we showed, yes, also, when we inject into the mice and treat it with the neem bark extract, either by gastric gavage or by intracranially, we actually pre-incubate the virus with the neem bark extract. We see that MNB restricts viral replication, neuroinflammation, and associated pathogenesis. And that helps us to understand that Bithanolic neem bark extract prevent viral antigen dissemination, microglia-mediated neuroinflammation, and protects mice from the myelin loss. So that actually, we wrote a paper in 2019 that which is published in 2020 in Frontiers in Cellular Neuroscience, and the home message is MNB restrict viral-mediated cellular entry. Methanolic neem bark extract restricts viral-induced cell-cell fusion. It inhibits spread and replication, and it also induces neuroinflammation. Then, with the help of the Dr. Parna Gupta and her student, Dr. Vishnu Das, we tried to understand what is the bioactive compound, because, neem bark extract has lots of other compounds, and maybe there are some steroids. So what we did the fractionation by polarity, and we observed there is a fraction called DCMF1, which can dimethyl chloromethane, when it actually inhibits the viral replication, viral fusogenicity, and when we try to understand what is this compound, we observe it may be a mixture, but the DCMF1 has a lots of part is nimbin. So that's what we try to understand, that how this nimbin or its total nimbuck extract can restrict viral replication. And as I told, that time we got the in the US virtual grant to work on the Swedish strain as well as in US strain to understand whether it can restrict a human with coronavirus. So Professor Debnath Pal from IISc Bangalore, he is a bioinformatician. He took the challenge because he is also a part of our consortium, and he showed that NEEM compounds dock near spike and ACE2 complex, which is a receptor for SARS-CoV-2, and RNA-dependent RNA polymerase, which is important for viral replication. So in one hand, MNB methanolic nimbuck extract can bind to the spike protein and can restrict the viral entry. At the same time, it can bind to the RDRP and restrict the viral replication. Mariani and her team at University of Colorado, they showed in SARS-CoV-2 US strain, when we are pre-incubating with the methanolic nimbuck extract, it can reduce the cytopathy and can reduce the viral replication. So that is one of the studies we wanted to see, that how MNB interacting and restricting SARS-CoV-2 infection. And we published this paper from the consortium work, which is published in Virology, and it got the cover page. It mainly shows that MNB binds to the ACE2, which is the receptor for the human coronavirus SARS-CoV-2. NB can restrict the initial entry cell-to-cell fusion. It can interact with host cell receptor, and it can reduce the replication. So while this work is going on, we were very much interested to see, okay, that is about the beta coronaviruses, but can it be anti-cancerous property also? So one of the PhD student, Mr. Shaurabh Kumar, now he is Dr. Shaurabh Kumar, and he is a postdoctoral fellow at University of Oklahoma. He took a lot of biochemical approaches, cell biology approaches, and he showed that MNB has low or no cytotoxicity in normal cell, but it can selectively kill cancer cell by apoptosis. It can cause cell cycle arrest, so it can reduce the cell division, it can inhibit the cell migration and epithelial to mesenchymal transition, which are the important for the metastasis of the cervical cancer. And that work he published in BMC, Complementary Medicine and Therapy. Then he further understand what could be the fraction. So that's what he did mainly, anti-cancer property of methanolic nimbuck extract. To understand the mechanism, he looked at the P65 and P53 and actually activate the NF-kappa beta pathway, and NF-kappa beta pathway aids to the cancer cell growth and survival and promote P53 that induces apoptosis and cell cycle arrest. So in that way, we can say that P53 actually increases apoptosis and cell cycle arrest, and NF-kappa beta can reduce the growth and survival of the cancer cells. So then we looked for the anti-cancer property of methanolic nimbuck extract in apoptotic metastatic cell line ME180, and we showed that fragment is the most active fragment because he did the fractionation, and by looking for the bioactive compound through fractionation, we have the protocol in the paper. If anybody is interested, we can show that. And we did lots of flow cytometric assay to understand if to reduce a cell cycle arrest, if to induces apoptosis in cervical cancer cell and help to kill the cancer cell, if to induces oxidative and ear stress, which is actually one of the cause for the cancer cell to propagate faster. And also we showed that cancer cell are getting dying due to huge oxidative stress. And then he also took a approach of the spheroid, which is much closer to the tumor. That time we did not have the in vivo nude mice model, but now we have. But he showed that this spheroid culture also getting highly affected due to the F2 fraction. And that he highlighted in his paper that among the 9 fraction he isolated, F2 is the most potent in anti-cancer activity, and F Hello? Hello, can you hear me? Hello? Hello?
We are not able to.
Hello?
We are
Yeah,
not.
can you hear me?
We, we can hear you.
Hello? Yeah. So among the 9 other fraction, F2 was the most potent in anti-cancer activity, and F2 successfully inhibits the cancer cell progression by cell cycle arrest. And also he showed very elegantly that, just not showing, that F2 reduces ear and oxidative stress and majority in ROS dependent ear stress pathway. And HeLa spheroid treated with F2 fractions had decreased the viability the growth. And in that way, he actually showed that he published this paper in a limonoid rich in a cancer, BMC cancer, that limonoid fraction has anti-cancer properties, but it is ROS independent.
Dr.
And then
Jayashree,
we can say now
Dr.
that
Jayashree,
methanolic nimbark extract, while the Nimin fraction in mainly restrict the beta coronavirus entry, spread, replication, and finally reduces neuroinflammation by reducing the cell
Dr.
cytopathy. It also has the another fraction, limonoid, has anti-cancer efficacy by induction of apoptosis, cancer cell death, proliferation, migration, and metastasis. So
Dr.
this
Jayashree,
isolation of limonoid compounds from Nimbach actually helped us to do that. So now Miss Purba Bera, she is a PhD student. Now she is actually working on understanding what could be the other fraction that may also reduce the antiviral, can have antiviral and anti-cancer property.
Dr.
And
Jayashree,
now if I combine my studies, we have that salanol 3 acetate,
Slides are not
which
moving.
and salanol 3 salanine, these compounds, while salanine has low cytotoxicity in cancer cell death is aggravated, inhibit cancer cell proliferation, inhibition of cancer cell migration, and elevation of intercellular ROS level in cancer cell. Salanol 3 acetate in antiviral properties show less cell cytopathy, inhibit host cell entry, reduces viral infectivity, suppress viral gene expression, mitigate oxidative stress, and reduces inflammatory stress. So we can see that Nimbak extract and its bioactive compound has therapeutic applications. And when we did this work, when SARS-CoV-2 came, we are like got several media coverage, not only from India, like News 18 or News from India, Express Pharma. We also got several coverage, media coverage in University of Pennsylvania, Medicine, and also Economic Times, and from Colorado. So our major contributors is Dr. Lakish Sharkar, who is now a research associate at Mayo Clinic, and
we have communicated to her that her slides are not moving. We are just trying to get reconnected to her. you,
We are trying to connect with Dr. Jayshree. In the meantime, we have Nisarga Biotech team with us, so we would like to give their pending award. The award for Outstanding NEEM Innovation goes to Nisarga Biotech Private Limited. For receiving this award, I would like to request Mr. Tanmay Soman to please come on the stage. I request Dr. Vyas to please present this award to Tanmay Soman. I also request Dr. Venkatesh to present him with citation, certificate and memento. In recognition of Nisarga Biotech's visionary leadership in transforming the village pharmacy into a global medical fort and bridging centuries of Indian heritage with cutting-edge innovation at UT Health San Antonio, USA, you have achieved the prestigious milestone of obtaining both Indian and US patents for your novel neem leaf extract. This honor celebrates your breakthrough success in the clinical landscape of head, neck, and colorectal cancer treatment. Your commitment to scientific excellence and international collaboration sets a new gold standard for the nutraceutical industry, proving that nature's wisdom, backed with rigorous research, can redefine the future of global healthcare. Thank you, Nisarga Biotech. Big round of applause for Nisarga Biotech. I request everyone to please be patient and be seated as we try to reconnect with Dr. Jayshree.
Dear colleagues, there is a connectivity problem for her to get reconnected. Sorry for losing contact with her. But my request in the audience, if anyone who is, what do you call, aware of the science and the same subject which she has been discussing, can add some more points and enlighten us like Dr. Anna or somebody else. Because I know I'm sure chemistry knowledge is very good. Can I add some more to think? It'll be interest to the audience, please.
though we have been planning to have this sort of an interaction with the pharmaceutical side also for NEEM, this is the first time we have got a very good research-oriented paper on the application of NEEM in various disease control, especially the cancer control. And it's a very interesting topic. Unfortunately, due to the connectivity, we lost our lecture. And I would request whoever can add value to this will be interested to know more about it. I think Dr. Annadurai is coming, and one more hand has gone up from our. Yes, 1 second. Yes.
See what Dr. Balaji and Dr. Venkatesh was mentioning. See, there is a new dimension added to NEEM research. You know, I now call it as NEEM science and innovation. So a lot of things are happening. Those days, people are only focusing on azadirachtin, and the US Department of Agriculture has heavily invested and then innovated what it can do for anti-cancer activity, and they dropped it. It has no anti-cancer activity as far as azadirachtin is concerned. But you know, the non-azadirachtin component, like you know that last speaker was mentioning, selenin, nimbin, nimbandiol, diacetyl-nimbin, all those molecules have been shown to perform in certain cell cultures. At an organism level or at a rat model or something like that, it has not been demonstrated. These are all cell culture based studies, and they also throw some light that these compounds have some activity, and few individuals from the J.C. Bose Institute also, last time I met them, they are also heavily investigating on anti-cancer properties of the non-azadirachtin component, but other terpenoids. There during the COVID time, there was a lot of noise about the bark extract being used as a COVID prevention, and they were using only crude extracts. They used to boil bark and then drink that extract for 4 weeks, 8 weeks, like that, and people have reported very good recovery. And that paper deeply delves into how this NEEM bark components help in that. So there is a lot to be investigated and a long way to go, and it opens up a new Pandora box. We have been looking at only azadirachtin as a anti-pest agent and mosquito repellents and so on. Now the new dimension is added in the medicinal properties of these compounds. So it's a very interesting paper. Unfortunately, the line got cut off. Let us see whether we can ask her some questions. Thank you very much.
Thank you very much for adding some more interesting points. Tanmay wanted to, please come, take the mic here. It's from Nisarga.
Hello, am I audible?
You are audible.
Hi, so my name is Tanmay Soman, and I'm from the team Nisarga Biotech. So we have been working extensively with the doctors. So if you go through your bag, you will find a couple of products that we make. So when we speak with the doctors, we work with the allopathy doctors as well. So what they say is that we don't have knowledge about the neem herb or any other herb that you're talking, and in principle we cannot write your medicines because they are not clinically validated. So my suggestion to all of you would be, we need to back our herb with a lot of study and a lot of scientific data, and we need to integrate the modern science with the knowledge of Ayurveda. And we all need to come together, or else we are going nowhere. And with these few words, I would like to and I would also like to know view of Dr. Vyas and Balaji sir and Venkatesh sir on this. Thank you so much.
Thank you, Tanmay. Very valid. Validation is the most important aspect. Having a way forward in our life also will handle the validation part scientifically is very important. Anybody else is there who can contribute some more? Okay. I said thank you very much, Patanmai. Yes, sir. Madam, please come. Doctor, I forget your name. Come, Dr. Anuradha, please come. Please come here.
So I'm Dr. Anuradha Retakula. I'm here listening to the talks, and now I feel very happy that we have a platform where we can communicate between the corporates and the academicians. It is something that we lacked all these years. I'm very, very happy that the World NEEM organization has initiated such a nice event, an annual event, where we all come together and we discuss. So as a science communicator, I'm doing on a freelance basis. I'm trying to, communicate between different academic institutes and colleges, universities, all talking only about NEEM and how NEEM, in fact, encouraging the next generation to take up NEEM science, like my professor keeps saying, that you have to encourage the young generation and make them believe that there is a future in NEEM. And no one else can do better than all of us together, put together. So having said that, I would always think that there should be more of interaction between the the industry and the academicians, have some real research-based goals while developing the products, and such initiatives should be developed or initiated and encouraged, and young people like me and some others who have already been part of this kind of a journey will be happy to be able to be part of such a program, and some kind of a funding, some kind of initiations, everything has to happen. So these things will, yes?
So
Yes,
I
yes,
think you
okay,
have seen
yes,
a
yes,
little the
yes.
antiviral
So yes,
properties, so
yeah,
I
please
can
go
start from there.
Thank you very much, Dr.
So
Jayashree.
tell me when I can start.
Are you able to hear
Yeah,
us?
thank you.
Dr. Jayashree, are you able to hear us?
Yeah, sir, I can hear you. Can
Yeah.
hear me, sir?
Sure. Yeah, thank you very much for getting reconnected.
So
I think you can go through some more.
thank you, thank you. Sorry, sir, sorry for this.
No,
So,
no
sir,
problem.
I think I have already conveyed the message that Nimbark extracts and its bioactive compound Nimbin can restrict viral replication, viral spread, viral entry for human beta coronavirus SARS-CoV-2 as well as murine coronavirus MCoV. So now we are trying to understand which is the bioactive compound which may restrict the viral replication further. So while we are doing that, a talented group of students also started working on the cancer field. And Saurabh Kumar was the first student who took the challenge, and he wanted to show whether methanolic Nimbark extract can restrict the cancer cell propagation, cancer cell migration, and its property epithelial to mesenchymal cell transition. So he did a series of biochemical assays with flow cytometry, Western blot, as well as cell biology experiment, and he showed that methanolic Nimbark extract has low or no cytotoxic effect on normal cell, but it is significantly killing the cancer cell. And it inhibits cancer cell progression by cell cycle arrest and inducing apoptosis by reducing cell cycle A, cell cyclin A, cyclin B, cyclin CDK1, NF kappa B, BCL2, and survivin. And he also showed that it inhibits the cancer cell migration. And one of the property of the cancer cell to migrate is epithelial to mesenchymal transition. So that paper he published in BMC Complementary Medicine and Therapy, where he mentioned that methanolic neem bark extract, stem bark induces cell cycle arrest, apoptosis, and inhibit the migration of cervical cancer cells in vitro. He did further lot of studies to show due to time constraint, I'm not going all through, but he showed how it causes restricts apoptosis. So he showed that MNB inhibit NF-kappa beta pathway, which aids in cancer cell growth and survival, where promotes P53 upregulation. That induces cancer cell apoptosis and also causes cell cycle arrest. While he was doing that, he also wanted to show whether this can have anti-apoptotic properties. So he did several studies by showing the cell cycle arrest in S to G2 phase, and also he showed that by ICF it increases apoptosis. But one of the mechanisms for apoptosis is reactive oxygen species induction. He showed in his studies that no, Nimbuk extract do not restrict reactive oxygen species, rather it actually helps in the NF-kappa beta pathway, which is one of the cornerstone of his studies, which made a significant contribution in the field that generally we think like we can reduce the oxidative stress and can help to cancer cell to die, but his studies said no, this is not the case. And then Saurabh wanted to study whether he can find the bioactive compounds. So he did the fractionation, and for this fractionation, our postdoc, Dr. Sharna Gaja, she had lots of experience, and she actually helped in our antiviral studies. She helped Purbabera and other people to work on the fractionation, and we took the lead from there, and she had expertise to fractionate, characterize. So she characterized salanol 3 acetate and salanine. And Saurabh did actually this methanolic Nimbuk extract compound to show how it reduces the apoptosis. And he also took another cell culture model, which is a in vivo, in between in vivo and in vitro model, that is a spheroid model. In that spheroid model, he showed that when he used different concentration of the NEEM product, like 5 to 25, 50 and 100, there is a significant reduction cell death of the by apoptosis that actually showed that yes, this product has ability to reduce the can kill significantly the apoptosis. And this work he highlighted that among the 9 fractions he fractionated with the help of chemistry people and Dr. Sharna Gaja, he showed that F2 was the most potent in anti-cancer
Sorry to
activity.
bother you. Can you wrap it up,
Successfully,
please?
yeah.
5 minutes.
Yeah, sure. So this work was published in BMC Cancer. So now we can say that Nimbuk extract has pleiotropic role both in anti-coronaviral efficacy and providing anti-cancer efficacy. And now Purba is trying to isolate the fraction, and our study showed that in cancer, salanine actually one of the bioactive compound which may restrict viral cancer cell propagation, whereas salanyl 3 acetate, which Dr. Sharna Gaja actually isolated, and she is in the audience also. She showed they are Miss Purba Bera is showing it has coronaviral properties. And due to our cornerstone studies, our study got several media coverage, especially when due to the SARS-CoV-2 from Economic Times in India, several other News 18, Telegraph Online. Also, we got several international coverage in Asian scientists and University of Pennsylvania. And the people significantly contributed are listed here. Due to time constraints, we are not going to discuss that, but this Nimbuk study made a significant impact in our overall growth of the lab. And I showed some of the data of some of the students, which I already showed, Lucky, Debina, Purba, Sharna, and Saurabh. But there are other people also who are working in different aspects of the proposal. 21 students already graduated. 15 students are currently pursuing their PhD in my lab, and we are having a good group of collaborators. We are gifted with the good collaborators who help both in our NEEM studies, and I am funded by several funding bodies, and I'm thankful to Pfizer Kolkata for giving me an opportunity to have a good group of students and the facility, and several funding bodies like SCRB and DST, CSIR, DBT, and also from National Multiple Sclerosis Society, US, ASEAN, American Society for Microbiology, and National Multiple Sclerosis Society. And this is my talented group of students, and thank you all for your patience and for giving me an opportunity to present my work.
Thank you very much for reconnecting and concluding it. Thank you very much. Very interesting study on this. Hope we'll be getting more research articles as we go by. I think now we
Yes.
have any questions, answers? Yes, we have Dr. B. Anuradha. One question, madam, please be online.
Okay.
We have one question
Yeah.
for, yes.
Good afternoon, ma'am. I'm Dr. Anuradha. I have worked on NEEM, but using an insect model system. So my question to you is, you said there has been a significant decrease in the ESM transition while using the neem bark extract, I believe. So have you also looked at the cytoskeletal level, if there has been any?
Yes.
I would be interested to know on that. And my second question is, you said there has been a decrease in the gene expression of cyclin A, B, CDKs. Have you also looked at cyclin E? I'm basically trying to understand if it is just the G to M transition that has been affected, or even the G1 to S transition is also affected. Because there seems to be slight difference in the model that I have used and the model that you are using. So I would like to understand that part of it. Thank you.
Yes, thank you. These both are very intriguing questions. And answering to your first question, yes, they also alter cytoskeletal changes, like especially the microtubule. In our virus model, we see there are lots of microtubular changes. In cancer, we didn't particularly look at it, but there are proteins like ZO1, which are the structural proteins, and these are part of the cytoskeletal. There is an alteration, E-cadherin, N-cadherin, so there is an epithelial to mesenchymal transition, which is associated with cytoskeletal changes. So yes, Nimbuk extract, especially in viral model, we see lots of cytoskeletal changes that actually is also helping the virus to restrict their replication. That is one thing. And for cancer also for sure, because as we tested those like ZO1, vimentin, and these small cytoskeletal molecules. And now coming to your second question, as you asked, is that G2 to M phase, yes, we didn't see much arrest in the synthetic phase. There could be one reason that the choices of the cell, we took either tumor cell, that is HeLa, or ME180, which is cervical cancer metastatic cell line. So maybe depending on the cell line, it can also restrict the synthetic phase.
Thank you, ma'am. Thank you very much.
Thank you.
A very good afternoon. Ma'am, it was a brilliant presentation, very nicely, very convincing work done. I am Dr. Tarun Kant from Arid Forest Research Institute, Jodhpur. And my question to you is that, of course, as we know, I mean, in India we know it, that NEEM and its products are used because in Ayurveda also, and it's a village pharmacy, it's a miracle tree, because it has got antibacterial, antifungal, immunoboosting properties. And it has been used for all these reasons. And now your research convincingly also scientifically backed research results indicate that it has got anti-cancer properties, as well as it has got the properties for, I mean, anti-cancer is, I think, the major thing that you are showcasing in anti-SARS-CoV, the COVID viral, antiviral properties. Now, my question is that there is a paradox in the world that at one end India is promoting. We know it because we know it from ages culturally.
I can't hear you, sir. Can you talk a little louder?
Yeah,
I can't hear you.
Culturally, we know that what NEEM means to us. We all know that. But at the same time, particularly in the West, the European Union, the EU, the Food Safety, Food Regulatory Authority, as well as Health Canada, they don't allow use of neem and neem products, saying that neem has also got some hormone disrupting properties, anti-fertility properties. So this is it a myth? Or if not a myth, it's high time we have to come out with scientific proof. The kind of study that you are doing, I think we need to do beyond this. We have to prove or validate, or we have to refute this thing that is going around, that neem has also got an anti-fertility property, endocrine disruption. I think your group is the right group to carry out research on that, to prove it, and then backed by further clinical research. So I think that is my question to you. Is there anything being done on this line?
No, I can tell you, like when I wanted to do the patenting and there were lots of innovation came, and as you have seen, I have a good group of US people are in my group. So I never I never heard that it has anti-fertility properties. If you can share some of the article with me, maybe we can think about it. But tell you the truth, that is not a major concern. The concern is how you can prescribe someone, like say if you are thinking about the modern medication, like take the neem bark extract in a spoon, right? You have to have a delivery mode. And another major question, what is very pertinent, like I am showing, if I incubate the virus with the neem bark extract, then it restricts viral entry. But you know, in our brain, nobody does it. In our lung, nobody does it. So we have to have a proper way, either by nasal passage, like nasal spray, so that it can bind. So I think your question could be valid because you might have more literature. I never faced any question like challenges for that, and nobody asked me when I present even in the international meeting. But major concerns are two. One is how to avoid the steroids, because this is the oldest part of the tree. Because you see neem bean, neem leaf, neem oil, maybe you know they are younger product, but neem bark is one of the most conservative part because it is for longer time staying with the bark, like staying with the tree. So one of the concern could be how the steroid we can avoid. That's what we are doing, fractionation. So bioactive compound may be a good way to go. Second thing is we need to coordinate with some company who can make a spray from the bioactive compound. And third thing is how to commercialize this bioactive isolation so we can make a cheaper cost, because if you want to buy as a didactin, it is so expensive that nobody can afford to have that. But if you go to Bhargava's neem bark extract, you can get it in much cheaper way even through the Amazon. So that way, I think the research should go in that direction to how to do the delivery, how to remove the steroid, and how to make a bioactive compound which can be applicable as a medicine, which is not only a problem with neem, it is a problem with any ethnomedicinal plant extract.
Right.
So that is the concern, and I will look at the literature whether it has anti-fertility property. But if it is so, we can easily do it in mouse system. We can look for their estrous cycle. We can look for their ovarian status. We can look for their cervical status, and we can check the hormonal level through the biochemical assay.
Yeah, ma'am.
But you please write a mail to me
I will.
your name and email. I will keep in touch.
Ma'am, I will definitely do that because I feel it's an urgency, because the literature is there which says that, and it's really disheartening to see those kind of things. And I think your group is the right group which can do research on this. Maybe if this is true, that there is some kind of an element of anti-fertility or hormonal disruption thing there, then probably fractionation can prove which fraction is having that, and by eliminating that fraction and making the drug without those fractions which are interfering and are causing such things. So I will share the literature,
Absolutely.
and I think
Dr.
it's high
Tarun.
time that we should refute and come out with a solution to this, because
Thank you, Dr.
internationally
Tarun.
there is a problem. Thank you so much.
Dr.
Thank you.
Tarun.
Thank you.
But
With
this
the
part,
permission of the chair,
this,
with the
yeah.
permission of the chair, I will address this gentleman's
Yes.
query. See, those days, 1970s, when family planning was at full swing in India, G.P. Talwar and his group had published a number of, short notes and papers, which says, neem oil can be used as a spermicide and, sprays and all that for, inhibiting fertilization. And that became an inhibition for marketing neem in European Union, particularly UK and Canada. But now,
Australia also.
But certain countries, now started accepting it. Your remark, European Union, a lot of countries are allowing neem now. And, I have published a paper along with Dr. Anuradha. She will talk in the afternoon about that, where we have done extensive studies on how the azadirachtin acts at insect actins, and it is very highly specific only to insect actins and not to mammalian actins. So the target, 700 and more species of insects being targeted, how a biological system like a human system can, be safe. So that answers the question that this molecule is highly insecticidal and insect specific and not human specific. So it has been tested in human cells compared to, insect cell lines. So it acts only on insect cell lines and binds with, insect actins and not to human actins. Thank you.
This is very valid.
Thank you, sir. Thank you.
Reply, Dr. Annadurai. Thank you very much, because this, the spermicidal property of neem is being promoted in various fora, and that affects the industry in a very big way. Thank you very much, sir.
Hello.
Hello.
It's okay.
Dr. Jayashree,
Hey, I will yield to the youth
Yeah.
very soon. Thank you so much for being here. It's just a comment that I want to make, and not really a question. I believe that the point that was made earlier about us working together with the WNO, supporting this organization, we have a gem of a nucleus here. Everyone should be participating. Everyone should be financial members. I'm not soliciting for them. I'm just saying that if we don't put our money where our mouth is, if we don't put our support behind the organization that is here to provide an avenue for information for all of us to grow and learn, then we are failing ourselves. Number one, we need to keep records. My husband would be happy to hear me say that because he's beating me all the time about keeping records. But experiential records, what we know is happening, not proven scientifically, but for instance, balancing the hormones. I have had women who can't get pregnant go on 3-month supply of NEEM capsules. Then they can have a baby. In fact, even twins. They blame me for it. But we need to record these experiences, and we need to share this knowledge, okay? If we don't, we're failing ourselves. I beg you to please collaborate and coordinate our experiences and our everything that we have that can benefit this organization and each one of us. Thank you.
Thank you. I think, yeah, he has one. No, Tanmay has one more.
Thank you. Doctor, thank you for a very nice presentation. It was very interesting.
Thank you.
I am Uday Pilar from Sequoia Biosciences. I'm the founder, CEO.
Hi.
And you have preempted my question to an extent, but there has been a discussion in this room about the need for quality assurance of Ayurvedic products. Now, my question is, neem bark contains many active elements. It would be very good if you have done some work on individual elements which may have that kind of specificity in acting on specific problems for which there is no cure in allopathy. That also will enable Ayurvedic medicines to be marketed and tested and labeled properly. Because in India, anything is labeled as Ayurvedic does not really deliver results because these are not really very good in terms of post-harvest keeping quality, unless cold chains are maintained, unless quality of the product is maintained. And one really doubts, even with most Ayurvedic products sold in the country today, whether the prescriptions which are laid down in Ayurveda are actually followed. Especially when you are looking at plants, you are not looking at even the genus and species specifically. Anything which is resembling that is used and passes off as an Ayurvedic product meant for a particular treatment. So your research work, which is fantastic, if it can deal specifically with the active agents which are producing those results, then you could have a very good product with specific content specified so that adulteration and violation of quality in our country is avoided.
Doctor, we can, yeah, it's a very good suggestion. I think you can discuss with her offline.
Sir,
and
I
connect it.
just want to make a
Dr.
comment.
Dr.
Like,
Jayashree,
yeah, yeah.
other one more person is having a view to express. After that,
Yeah.
whoever has to discuss with you, they will connect with you offline so that we are not running short of time for the next lecture.
Yeah, absolutely,
Okay?
absolutely,
Thank you.
sir. Yeah, thank you. I wish I could have come, but thank you, sir, for giving me an opportunity. And I will now log off. And
Kindly,
anybody has been okay.
Kindly do not log off. There is one more
Okay.
person who wants to discuss with you.
Okay.
So, sir, this is not a question actually. So, we have been able to identify some bioactive compounds from NEEM. So, nimbine, salinine, and nimbolites are something that we are able to standardize, which come from neem leaf and bark extract. And I'm hearing about infertility. So, when you use the neem seed extract, that is where you get the azadirachtin. And that is a compound that may cause infertility. So you never know.
No, don't make a statement.
No, I'm not making a statement, sir. But we have been able to.
It will kill the industry. Please, don't do that.
No, I understand, sir. But then we have done a clinical study in Delhi for COVID using our neem leaf extract, and we have done human trials for that. And it was done by the Ayush Ministry.
That's okay.
Yeah.
Fine. Yeah. Any more suggestions? Yeah, Dr. Devendra Kumar has on you. Yeah.
I'm Dr. Devendra Kumar from PJ Margo. I want to know whether nimbin pure compound was more active or whole bark extract was more active for antiviral activity.
That's a very good question, yes.
Can you talk a little louder, sir?
Yeah. I want to know whether pure nimbin was more active or whole bark extract was more active for antiviral property.
Sir, whole bark is more active against antiviral properties because, you know, when we fractionated it by DCM, polarity of the solvent, the F2 fragment actually had other compounds. Only nimbin is not the compound. So it may be a combinatorial effect of other compounds. So that is also very important.
Yeah.
We should look for the synergistic effect rather than just nimbin by itself. So your question is very valid, sir. Neem bark extract is more potent antiviral than the isolated nimbin.
Yes.
Thank you.
Yeah,
Thank you so much.
Dr. Devendra Kumar has got a very valid point. Thank you, Dr. Devendra Kumar. But keep your question short so that
Actually, I am Dr. Deepankar ma'am from Fortune Biotech. I am agree with you, but few things I am not disagree. One thing you told that the steroids. See, I was working from last 3 to 4 years, except on the NEEM. So, what is the peculiar things I observed, the chemical motif? So, in majority compound is the limonoids. So it is so beautiful thing is that limonoids is still nobody has done the complete mapping of the limonoids. Understand? So even the compound it is in different segments. Suppose the maximum cluster of compound which is present in the seeds, that is also present in the leaf also, and even bark also, even root also. Understand? So thing is that which one is the focus? Which one you have to particularly you have to focus on? Your target specific. So here one slide I have noticed that in nicotinflovin, one compound you have. Understand? This nicotinflovin, if even leaf also, this is a flavonoid compound. The flavonoid and phenol is present in the leaf. Similarly, people has reported this is also present in the neem oil also. Yes. Then steroids, it is plant steroids that is present in leaf also. Even it is present in the oil also. Understand? Oil is present in the seed. Even it is also, this some compound is present in the bark also. So this is the beauty of the neem. This is the whole plant. Whole plant is contributing the chemicals. That whoever delve in this research, only they are also understanding that. Second thing, azadirachtin is a isolation azadirachtin. Azadirachtin also present in the leaf also, is a bark also, even the seed also. Everywhere it is present. But the quantity is specified. Where it is present more, where it is freely available, where it is conjugatedly compact. So these kind of things are there. So our target is there now, specifically which is the compound. Now you have done one step ahead, because in the bark, what the studies you have done anti-cancer activity, that is in the leaf and oil also they have reported. But you have done the excellent thing in the bark also. Now all this collectively, if you do that, now we have to segregate which compound is particularly responsible for that, which is therapeutically important. That is the time has come now to focus on this. Anyway,
absolutely, sir. I agree with you. And I hope I didn't make a wrong, it is not understood wrongly. I'm not talking about the steroid present in limonoid. But you see, neem bark extract, when you are taking, say one of the question is, say you are taking a place in a busy city, or you are taking a place in a serene place, you are taking from Africa or you are taking from India, we have to test the steroid. That's the thing left with our work. Because when people ask me the question, did you check the steroid level? I said no. So that's what I wanted to mean, that I have to do that.
Thank you, Dr. Jayashree.
Plant steroid is useful things. Plant steroid, phytosteroid, that is called the phytosteroid. So this is always useful. Thank you so much, everybody.
Thank you, Dr. Jayashree. It was a wonderful topic and very interesting, and it has created so much of interest among the audience also. We'll get connected to you offline on a later date. Thank you very much.
Sure. Absolutely, sir. Sir, log off now? Thank you, sir. Thank you. And thanks to all the organizers and all the participants. Thank you, sir. Thank you.
Now,
bye-bye.
Yeah, thank you.
Before that, I will make a statement about the comment made by Tanmay that, nowadays, lot of studies are done with the specific compounds. So when you do with the specific compounds, you know about the mechanism of action and purity, etc. That is one. Second thing is that our WNO is quarterly publishing the newsletter. If the people who are not the member, they should become the member and they will get the copy of the newsletter. In that, there is a list of several papers along with the abstract. So then you will know what compound for which particular purpose, and these are purely research papers.
Thank you, sir.
Now request Mr. Milan Mehta to honor our co-chair, Dr. Pushpavalli.
डॉक्टर जेकब मैथ्यू फिलिप के बाद हजारिया बाबू हमें ऑनलाइन ज्वाइन करेंगे ठीक है अनाउंस तो कर दूं कि लंच है उसके बाद टेक्निकल सेशन सवा दो पे 12:15 2:15 Thank you everyone for your patience and attendance. We would now like to invite you for lunch. It is on the second floor. I request everyone to please proceed for lunch. We will meet here again at 2:15 for technical session 2. We will have Dr. Jacob Matthew Philip with us from Oman, Dr. Azaria Babu, Dr. B. Gurudev Singh, and Dr. Balasubramaniam Vairappan. Thank you.